to be talking about attention today, which is, in my opinion, a really, really cool topic. How the brain does this is kind of amazing. We have tons and tons of information bombarding our senses at any given moment, and we have these mechanisms that can filter all of that information and kind of narrow it down to just the things that are important for our goals and our intentions and things like that. All right, so today's outline. So today we're going to look at how the brain modulates and interferes with these sensory signals that are coming in to kind of filter out that information so that it's only focusing on things that are important for what our goals and our intentions are. So first we're going to discuss what attention is and what the different types of attention are. This is still like really bugging me that it says slack in the middle of my screen. Let me see if I can fix that. Does that work? Nice. Okay, cool. So then we're going to start exploring different anatomical regions. We're going to look at how those different regions then kind of cooperate in these networks, feeding information back and forth to one another, how it ties into things we've already talked about, like the dorsal and ventral stream. And then at the end, we'll talk about how these attention mechanisms can fail, things like neglect. So really, really interesting disorder. People will just completely ignore half of the body, half of a region in space. Well, you'll see there's a picture. This guy has like half of his beard shaved and the other half isn't. He's completely neglected half of it. Really, really interesting disorder. So, attention is kind of spotlight that we have on things that are interesting or things that are relevant for our goals. William James is going to come up again when we talk about emotion. This guy was way ahead of his time. So this was 1890. There's a lot of work that he did on emotion that's really interesting and still relevant today. And this quote was just something that really captures a lot of what we're studying today about attention that he kind of put forth in a really coherent way. So everyone kind of knows what attention is. It's intuitive. We know that there's all of this information out there and that somehow our mind kind of is pushed in one direction or the other, right? And there's several different objects or several different trains of thought that are possible, but we're really only able to attend to some of them. So this kind of hints that there's these bottlenecks where we can't attend to everything. We have to attend to these these focused things, focalization, concentration of consciousness. And it applies that we have to withdraw from some of them in order to deal with others. So it shows these limitations of these systems. And it's a condition which has a real opposite in the confused dazed and scatterbrain state. So we'll talk about that in a minute when we talk about the global state of the organism. Attention is really, really good when we're awake and when we're alert, and it's not when we're kind of in this kind of dazed state. So I'm going to do this little experiment with you. So I want you guys to look at the middle of the screen, and then when I hit the button, there's going to be a bunch of letters that will pop up, and I want you to focus your attention up here and look for a word. And then I'm going to take it off really close, really fast. So, what did you guys see? Brain. Now, did you guys see science down here? No. So that kind of shows these bottlenecks, right? We have this spotlight of attention. So there's all of this stuff going on, but we're able to direct these processes to regions of space and ignore all of this other information. And so it shows that a lot of stuff is being lost, a lot of stuff is being filtered out. This was something that Hermann von Helmholtz did himself. He hung this sheet on the wall, wrote a bunch of letters on it, and then he used this spark where he would just like light up the room really temporarily. And he noticed that he could only keep in memory the things that were in his spotlight of attention. So he kind of decided ahead of time where he was going to look. It talks about how there's this top-down control of these mechanisms, right? And this was kind of the idea that everything that was outside of it was kind of filtered out and ignored. Oh, my animations failed. So arousal. So this was something that William James was getting at, the kind of global state of the organism. This is not sexual arousal. This term gets mixed up a lot in the literature. This term is used a lot in psychological science. This is something that's going to come up a lot when we talk about emotion. And it's talking about this spectrum that we can be on from deep sleep to hyper alertness. And attention mechanisms operate really, really good at the higher ends of the spectrum. So when we're really, really hyper alert, that's why we're so addicted to caffeine, because there are receptors in these regions that maintain attention that are really sensitive to these types of drugs. They allow us to focus in, but that also burns these systems out, and that's why you crash after like 2 hours of drinking a ton of coffee. So what I want you to know basically from here is that arousal is this kind of global state, and that attention's operating really well at the high ends of it. So if you're half asleep, you're probably not going to take much from this lecture. So there are different types of attention. We can have voluntary attention, which is referring to these top-down processes. So we're the driver, right? It's this controlled process. We're deciding what we want to pay attention to. It's directed by goals or intentions or expectations. It's guided by what's going to produce the best reward in the end. And this is an ongoing process. Our brain is constantly making predictions about how we can achieve these goals, and it's using these attention processes to filter out information that's specific to those goals. Whereas reflexive attention, or exogenous attention, is a bottom-up process. This is something that we don't have control of. This little marionette, like, do we have free will? And this is something that is reflexive. The reason the word reflexive is there is because it's something that automatically orients us to a stimuli. So this would be like a loud noise. If all of a sudden there were a bunch of people screaming outside, all of you guys would stop paying attention to me. Your goals would change. And this is a really important mechanism for survival in general, as we'll see on this next slide. So voluntary is we're the driver, we're in control. Reflexive is more of this. We've talked a lot about reflexes in general. It's this thing that we don't have control over. And as we'll see as we get later into the lecture, a lot of these reflexive processes are controlled by older brain mechanisms, these deep structures in subcortical regions. So these two things are working in opposition to one another. And it's this tug and pull between maintaining our goals, paying attention to what we want to accomplish, like you guys paying attention to the lecture right now, versus orienting to stimuli that are distracting. And this is important because if we're too focused on something and there's something really important happening outside, then your survival chances go down, right? You're missing something that's really important for you to be successful. Whereas on the other end of the spectrum, if we were constantly distracted by everything that came up, every little sound, every click, every whatever, we wouldn't accomplish anything. We would never finish any of these goals. And so there's this really fine balance between these two concepts. And they're constantly working against each other. So when reflexive attention comes on board, it shuts down voluntary attention, shifts it away. So most of what we're going to talk about through the lecture, we'll get into some of the more reflexive stuff, but most of the stuff that's actually studied in psychology is voluntary attention, because it's something that we can manipulate very easily. And there are two types of voluntary attention. We can either be attending to things overtly, and so this would be something that's very obvious to someone that's looking at you. So if you're orienting your head to look at something, or moving your eyes, or moving your head to listen to something more intently, this is referring to overt. It is conspicuous, it's obvious. Someone can see that you're paying attention to them. The other one is covert. And so this is something that's really interesting. We don't have to orient to be able to pay attention to something. If we want to eavesdrop on someone's conversation, we can do that without looking at them, without giving them any kind of indication that we're turning our head towards them, anything like that. And most of the research on attention studies covert attention because it involves these inward directed control mechanisms of attention. They can kind of filter out the movement and all of that kind of stuff because everything is going on inside your head. So No external clues for covert. It's inconspicuous, like this guy over here. And this was an important mechanism that had to evolve because we needed to be able to not give indication to prey that we were coming up or something like that. Cool. So overt, very obvious. Covert, not very obvious. Cocktail party effect. This is something that I hinted at when we talked about audition. And audition is this amazingly complex system where, like, let's say you're in this party, this huge party, there's music playing, all of this stuff. The actual waveform that's hitting your ear is extremely complex. It has all of these different sounds all mixed together. Yet somehow the brain is able to pick those things apart, and we can selectively attend to one conversation. So like all of this stuff just sounds like blah, blah, blah. This guy here, and it's cut off the bottom of my picture, I apologize for that, but there's a guy down here talking about a new sculpture at a gallery. And so this guy is pretending to pay attention to this, golly, that's great guy, but really he's listening to someone way over here. And so his brain is kind of tuning all of that stuff out. So we have this ability to selectively attend to any one thing at a time. Even though we have all of this sensory information bombarding our brain, we're able to pick out what we think is the most important for our given goals at that time. So this is something, if you were in this really like busy thing, so this guy is specifically trying to pay attention to this, but something like your name being said on the other side of the room will automatically attend you to that thing. So there's even things in your brain that even if you're not consciously trying to pay attention to things, it's still picking up clues that you're not paying attention to. And it will reflexively orient you to those things. This is something, I wish I had time to do this. There's a really good demonstration of this that I've seen in classes before where they have two people come up to the front of the room and they both read you a story at the exact same time. You have half the class pay attention to one and half the class pay attention to the other. And then you ask at the end, so if this half was paying attention to this one, you ask them what the other one said. And it's this demonstration that you can only really pay attention to one audio stream at a time. There's this bottleneck that kind of prevents us from attending to multiple things. You might be able to pick up on little bits of stuff coming from the other auditory stream, but if you're directing your attention to one and there's something else going on, all of this other stuff just gets filtered out, blah, blah, blah. So this is this really important note that there's a very limited amount of information that we can pay attention to, and it's usually one auditory stream. And so they do these studies where they actually put in auditory information in both ears, and they tell the subject which one to pay attention to. And the information from the other ear just gets completely lost. It doesn't get stored in memory or anything. So, bottlenecks. So all of this has kind of been hinting at this idea that we have this filter mechanism. We have all of this information bombarding us. We have billions and billions of bits of information hitting the brain at every moment, but we have this filter mechanism. We're only able to bring in a limited amount of information. And this is what gets to our higher order processing centers. But there's these questions, once we get into the research and we start looking at this stuff, there's this debate going on about whether or not this filtering is happening really early in the processing stream. So are we selecting things before it's been kind of built up into these like coherent perceptions? Or are we building perceptions of everything and then selecting at these higher order areas once they've kind of figured out what everything is? And so that's kind of this idea. So if attention is happening late, then that means all of the information is being processed. If the attention is happening early, that means we're getting rid of tons of sensory information before it even gets to any of the higher order stuff. So this slide, what I want you to get out of it is this top point right here. Bottlenecks are referring to this limited amount of information it gets in. It refers to the end of a bottle. You have a ton of stuff in the bottle, but only a little bit can come out at a time. And we'll go over these two questions on the next couple of slides. So I'm going to show you this. So this was a picture. It was like used in early psychology. It was someone spilled an ink bottle, and it's just kind of a mess of different blobs. And so when you look at that, your sensory information, your early sensory stuff is just processing all this black and white contrast, right? So I just directed your attention in a specific way. I told you guys that this is nonsense. Now, if instead I told you that there's a Dalmatian in the middle of the screen, and that this is a tree and this is a park, all of a sudden your brain is giving these early processing streams different types of information. It's telling these early systems to create different types of perceptions. And now all of a sudden this dog jumps out at you. And this picture, if I were to show you this picture 2 weeks from now, a month from now that I've told you what it is, this is going to jump out at you right away. We've already oriented your brain to recognize this and to recognize that there's a high order thing right here. And so your brain is automatically filtering that early stuff and saying, I want you to pull something coherent out of this. And so this is kind of getting at that idea of whether or not these things are happening early or late. And that's what this is about right here. So early selection is referring to this idea that the filter process is happening in these really early stages. So when I'm talking about early stages, I'm talking about primary cortices, right? So primary visual, primary auditory. We'll see later that this may even extend to subcortical, so even earlier, so thalamic nuclei. But this is this idea that there's something going on in the brain that is making it so that stuff really early on in the stream is getting filtered out. There are other researchers that think that it's more of a late selection idea. And so in this case, we actually have all of the sensory information is being processed, every single bit of it. We're forming coherent perceptions of every single one of them, but then our attention mechanisms will then select from those higher order regions what's important. So we'll look at evidence from here on out supporting one or the other. I personally lean a lot more towards the early selection from what I've seen. But something that's kind of nice about early selection is that you can still have late selection mechanisms built on top of it. So you filter out a ton of information and then you're able to filter that even more. But a lot of the evidence isn't pointing towards a pure late selection. So a lot of information is saying we don't really process everything and then select. We filter it really early on, and I'll show you that evidence here in a minute. So what I want you to take from this is that early selection is affecting primary cortice regions, primary auditory, primary visual. And it's modulating the activity in those early areas so that only the important information gets out. Whereas late selection is acting on kind of these unimodal association areas, the ones that have made more higher order perceptions from the early sensory information. So we'll get into, now that we kind of know what attention is and what the limitations of it are, we'll look at how cognitive neuroscience has started to pick apart these things. And I'll start with something that we mentioned on the first day of class. This was a task that was used by early psychologists. This is Dr. Posner again, a guy that is here at the University of Oregon. He started brain imaging and all kinds of stuff, really, really influential in the field. So he had this task where he was cueing attention, and this was all done covertly. So what the subjects do is they stare at this fixation cross and they don't actually move their eyes back and forth. They're just supposed to use kind of their peripheral vision. They're supposed to attend to things without making any obvious motion. And these cues that are used can either be endogenous, where it's an actual arrow that's pointing towards where the stimulus is going to pop up, or it can be exogenous, which is kind of this reflexive cue, something that pops up and tells them exactly where it's going to be. And this task can be used to look at spatial attention, so asking where the stimulus was presented, and it can also be used to study feature attention. So you can ask what color this stimulus was, or whether it moved, or whether it was a specific item, something like that. And early on, before we started looking at the brain, we had this indication that attention was really important in speeding up the processing of these things. So reaction time and accuracy was better. So they were better at saying what color it was, and they were better at saying where it was if the attention was directed in the right direction. So in this case, this B situation right here, the attention cue was pointing in the wrong direction because the stimulus actually ended up on the left. And so in a trial like that, their reaction time would be a lot slower, and they'd be a lot less accurate at saying where that thing had actually popped up. And so this was a task that was developed really early where they only used these kind of things, but this kind of a task, it's not exactly the same, but this is something that is used in brain science now to actually explore these mechanisms. And so real quick on this next slide, I want to show you kind of the anatomy of attention. Don't worry too much about memorizing these brain regions or anything like that. That's not what's important. The idea is just kind of understanding that this is a complex process that kind of involves a lot of different regions. And there's both cortical and subcortical here. So that's kind of what you see here. So everything in the pink brain is cortical, and then you have this blue thing is the thalamus and some of these midbrain structures like the superior colliculus. The reason it's blue is because it's just highlighting that's deep in the brain. So cortical, you have these regions, like I said, don't really memorize what these are, but you have stuff like in the frontal lobe that's responsible for orienting our eyes. You have stuff in the posterior parietal lobe, which is back here, that's really important with determining where we should attend to. Posterior superior temporal cortex, that's this stuff down here. So this is feature selection, so deciding what types of items to attend to. And then cingulate cortex isn't on here, but that's kind of what rides along the top of the thalamus right here. And that's what it's kind of a super highway of information. It connects a lot of these different systems together. Something I want you guys to notice on these cortical maps right here is something that we talked about in object recognition. You'll notice that this pathway right here, down on the bottom, has a lot of stuff to do with features and objects, identifying things. This is the what pathway. You'll notice that this one that goes up towards the parietal lobe is talking about spatial relationships. That's the where pathway. And so these attention mechanisms are taking advantage of what the brain is already doing in identifying things and figuring out where they are to use that information to direct attention. So this isn't a new idea. This is just showing you that what we've already talked about is kind of being elaborated on with some of these frontal lobe mechanisms that direct some of this stuff. Subcortical stuff is referring to like thalamus and these other midbrain structures, and these are important for reflexive type attention, for orienting us to things. So superior colliculus is something that actually came up when we were talking about sensory information. This is where a lot of the auditory information will synapse at first before it goes to the thalamus. And so this region is actually processing that auditory information really, really early on to determine whether or not something's important. And that's what's controlling a lot of our orienting reflexes. And then there's also stuff in the thalamus, this Bolton nucleus. So like I said, don't memorize all of this stuff. This is just to kind of put all of the pieces together to show you that this is kind of a broad network of regions that are all kind of working together on this. So what we're going to be, this isn't an important slide for notes or anything. This is just to kind of tell you that attention is operating on all of the sensory modalities. Yeah. This is just saying, like I said, attention is operating on all the different senses, but a lot of what we're going to look at from here on out is vision, because it's the most widely studied and it's the most easy to study as far as attention goes. So that's just to say, like, we're going to talk about a lot of vision stuff, but keep in mind these same processes are happening in the different modalities as well. So this is to demonstrate what we already know about sensory stuff, to show you how this changes when we add attention to the mix. And so when a stimulus is shown to a person, so this is a visual stimulus, when EEG is being done on the occipital lobe, which is where the visual cortex is, you see this big positive ERP. So ERP is an event-related potential. So this is so you average across a ton of different trials, and you see that this wave is similar in all of the different trials that we see. So this is a sensory signal. This is something we see very early on. It's saying, I just saw something. This N1 wave is not as important. I want you guys to kind of focus on this first deflection right here. This is confusing. I don't know why EEG people thought this was a good idea to put positive on the bottom and negative on the top. But that's like a common thing in EEG literature. So don't be confused by the fact that this says big positive wave and it's on the bottom. But the important thing that I want you to know here is that there's this big wave that we already know about when people are looking at something. That's going to be important on this next slide. Because attention affects that wave. And so what attention is doing is it's modulating the amplitude of that wave. And I'm just having you focus on the P1 wave. It's also affecting the other waves as well. But what you can see is that when we have them actually attend to the stimulus instead of attending somewhere else, that wave that we see for sensory information is way bigger than it normally is when you're just looking at something passively. So when you have these goals and you have these attentions, somehow, and I think this is like freaking amazing, somehow our brain is boosting the signal in these early visual areas. It's saying this is important, make this signal stronger. And so this dotted line is the stimulus when they're just casually looking at it without any goals or intentions or anything like that. And then once they attend to it, it's this dark red line. And so there's something going on that's just boosting these early sensory signals. And so this is evidence for these early selection methods. And so this is one of those mechanisms that's being used to try to boost up the signal of the things that are important so that they kind of rise up above the noise of all the other sensory stuff. And so before any of this stuff is processed, somehow the brain knows what early stuff to boost. That's what's really like kind of amazing with all of this. Like the brain already knows what types of things on the early maps are important for the types of objects that it's looking for. So this right here, EEG, is summing up thousands and thousands of cells, right? But this was a really, really cool indication early on that we were seeing early selection. And so you had these researchers then that wanted to kind of expand this work and see whether or not we were seeing this at a single cell level, or whether this was just something that was kind of a summation of all of it. All right. So they took monkeys and they did single cell and multi-unit recording on them. And they recorded from neurons in V4. So V4 is one of the higher order visual areas. So you'll see like V1, V2, V3. V1 is primary visual. So this is after things have already been processed ahead. And so they showed them a bunch of different stimuli until they worked out what these cells were kind of specifically tuned to. And this cell in particular was really liking a specific color, so it liked the color red, and it also liked that color red to be horizontally oriented. So they knew that this cell was really, really active for this red cell right here, for this red bar horizontally aligned right here. We know though that these cells in these higher order visual areas, they have bigger receptive fields. And so they're not just looking at a small region of space. And so they were able to put this green bar in the receptive field of that cell. So the cell was seeing both of these things. And that's really important. Because what they found was that this neuron was firing way more action potentials when they were attending to the preferred stimulus versus when they were attending to this distractor up here. So you can see that this red bar is still in that cell's visual field, and so it's still firing action potentials. But when it's attending to it, those signals are really, really boosted. So this is more evidence for that early selection. Because they also saw this, they did further work and found this all the way back as far as V1, so the primary visual cortex. And so there's something going on, and we'll look at some of the main contenders for who may be doing this in a little bit. But somehow there's higher order brain areas that know what they want to look for and are sending signals to these early visual areas to boost signals for those things that they're looking for. So more action potentials when something was attended. This next one is looking at evidence from FMRI. And so keep in mind, this is looking at thousands and thousands of cells. So they used a queuing task. So there was an arrow that showed them where they should be looking. And then 8 seconds later, they saw this flickering black and white checkerboard pattern. And they were asked where they saw the color gray. I think I'll see that in a second. So this was an important point. 8 seconds, this delay between when they saw the cue and when they saw the actual stimulus is important for MRI, because MRI, HRF stands for hemodynamic response. That is the flow of blood into that area. It takes time. And so they had to separate these two things because they wanted to be able to tell which was for the cue and which was for the actual response. So subjects made a response if they saw any gray targets on the cued side. So if they saw anything gray in the direction that they were supposed to look, then they would respond, yes, I saw it. And what they saw was that there was greater activation in the contralateral visual cortex. So contralateral just means that, like, if they're looking at the left, then the right half of the brain is lighting up, and we know that that's where the information is being sent from the left visual field. And so this is evidence, again, that we have these boosted signals that now we're picking up on MRI. This is pointing out the fact that these are higher visual areas. So MRI might not be good enough at picking up whether or not those boosted signals are happening early on. But this is kind of evidence for late selection as well. So we're seeing really boosted signals here. So you can see there's like no activity at all on the other side that they're not attending to. Even though there's something there, the visual field is picking up stuff that's being shown on the other side, but the activity is overwhelmingly greater on the side that they're attending to. And this is attending to the right, so that's when they're attending to the other side. There's no activity on this side. So, really cool demonstration of selection also happening in these higher order visual areas. So this is where I kind of think things are going, is that signals are boosted in these early areas that filters out a lot of information, but there's still quite a bit still coming through. And so attention processes are still working to select higher areas as well. I'm more skeptical of the fact that, like, late selection is happening on its own. We see, like, the information is still getting through, but I don't think it's purely late selection. I don't think all of it's being processed. So this was something that out of this work was this theory about what's going on. And it's this idea that when different stimuli are falling within that same receptive field. So this idea was that if two things fall within the same receptive field, there's kind of this competition going on between them, and attention needs to favor one or the other. And so the signals will kind of be even with one another, and then once attention gets involved, it'll boost the one that's more important. And so as you move to higher order areas, the receptive fields are getting bigger and bigger. So we talked about that when we were talking about vision. So in visual cortex, those cells are responding to a very small region of space, but as you move to higher order areas, those cells are responding to large areas of space. They're firing for an entire table or for a face or something like that, not just for a specific region. And so when you get up to these higher areas, there's multiple things that are all falling within that same receptive field, and it takes attention to kind of resolve that competition between the two of them. And this next slide is going to kind of show some proof of that idea. So this was some evidence from MRI. Sabine Kastner is someone who comes up a lot with attention work. She was a really big, influential part of the field. So they had this paradigm up here where they were either showing these kind of nonsense objects one at a time, or they were showing all of them all at once. And they had people go through and just look at them passively without any goals or intentions to try to see what the brain was doing without attention. And then they had them go through and actually attend to a specific one. And so what we saw with just the passive viewing, this was when attention wasn't involved at all. You see here in this higher, so this is V1, they weren't able to pick up on it in V1, and that might just be kind of a limitation of MRI. But in V4, in this higher order visual area, when they were showing things sequentially, that's this first column in these two boxes on the left, you saw the signal was boosted when there was just one thing on the screen. But when there was more than one thing, those signals were competing with one another and the overall activity went down. And then when it went back to just one thing being showed, the signal went back up. So that's important. They know what the brain looks like when it's only attending to one thing versus what it looks like when it's attending to a bunch of different things at once without any type of goals or intentions. So then they had them direct their attention to one of those specific objects. And what they saw was that this competition went away and the signal looked just like it did when there was only one thing on the screen. And so it was boosting the signal for the one thing that it wanted to attend to and kind of ignoring the rest. So they think that this was the competition kind of being resolved. It was like, I don't care about all four of them, I only care about that one. And so now you see a signal that looks more like object recognition when there's only one thing on the board. So this was kind of this idea, the effect was the largest in V4, which is one of these higher order processing regions. We don't know because MRI doesn't have really good temporal precision and things like that. We don't know whether or not this is occurring in V1. But this is just that idea that we're seeing this boost in signal. And this slide in particular is trying to get at that last slide that we just talked about. Is this reduction that we're seeing right here because of the fact that these things are competing with one another and it's reducing the overall signal? And because we've attended to something, that competition has been resolved. So we'll shift now to subcortical stuff. So these researchers, they discovered that these early processing regions, so like early visual cortex, had this effect, and so they're like, well, is this happening before it even gets to the cortex? Is there some kind of signal that's being modulated early on in the thalamus? And so what they looked at, these early structures have a really easy to map out wiring pattern. They can tell exactly where the input is and exactly where the output is. And so they found that there's a lot of connections from V1, so from visual cortex, back to the thalamus. And they think that this might be something that's involved with these attention mechanisms. So not only is the thalamus feeding information to the visual cortex, but the visual cortex is giving it feedback about what it wants. And these ones that are coming back from V1 are actually synapsing not on the processing region that processes visual information. So lateral geniculate nucleus, that's the one we talked about in vision. That is the nucleus in the thalamus that processes visual information. These axons that are coming back from primary visual cortex are synapsing on this area that surrounds that area. And that's kind of important for what we'll talk about on this next slide. And so they found evidence that there was differences in these LGN neurons, in these lateral geniculate nucleus neurons, when attention was involved. There's evidence that attention was affecting things even earlier than we had already assumed. And so this was evidence that we found from MRI before we started to explore this in the monkeys. So this was very similar to one we talked about earlier, where there were these checkerboards on the left and right, and they were supposed to attend either to the left or right. And what we saw was that there was activity in the thalamus, just like what we saw on that earlier slide. There was a boosted signal in the thalamus, depending on which side they were looking at. And so it may be evidence that it's happening really, really early on. And this is that contralateral side of attention. So if they were tending to the left, then we see a boosted signal at the right. Oops, this is backwards because it's an MRI thing. But if we're looking at the right, then we get a boosted signal on the left. That's kind of the idea. That's where the information is being fed to. And so we have this evidence now that there's this early stuff going on. And so these researchers, they wanted to see what's going on in animal models because we have a lot better precision with these like single cell recording techniques that we have. And so this one is kind of complicated to really work out. So just kind of stick with me. And so these monkeys, they were trained on an attention task. They were trained to either attend to one side or the other. And researchers were recording from the thalamus neurons, and they were also, so the LGN, lateral geniculate nucleus, that's the actual visual processing in the thalamus. And they were also recording from those neurons that were surrounding the LGN that we talked about a minute ago. So these are the ones that are getting information back from the visual cortex. And so what they found was that activity increased in the thalamus. So this was consistent with what we had seen in primary visual and all of these other areas in the cortex. We had this boosted signal. But what they found was that there was decreased firing in those cells that were surrounding that nucleus. So the actual one that was processing information was getting boosted, and all of the cells that surrounded it were getting depressed. They were turning off. And what researchers have kind of come to the conclusion of is those cells that surround the lateral geniculate nucleus are inhibitory. And so they're actually depressing signals. And so when we turn off inhibitory neurons, it boosts the signal in the LGN. There's a lot going on there that's kind of hard to wrap your head around, but that's what we see in this graph over here. So this one on top is the lateral geniculate nucleus. So this is the one that's actually processing the visual information. And so when we're attending to it, this dark green line, we see a boosted signal. These on the bottom are the cells that surround that lateral geniculate nucleus. And so what we see here is that when we're attending to something, these signals are actually going down. And so these cells that are surrounding the lateral geniculate nucleus are inhibitory. And when we turn off inhibitory neurons, it allows these ones to be more active. All right. I love this picture. Like, go dad. Dude's just like looking at his phone, no idea. So, reflexive attention. So, this is this idea that we have these automatic orienting type processes. So, automatic grasp of attention. And the more salient a stimulus is, so the word salient means the more important it is, the more it stands out from everything else, the more chance it is of it grabbing our attention, the more likely it is. So we have this thing like our head automatically turns towards it, our eyes are oriented toward it, our arm goes out so our sun doesn't get clobbered. Now this is something we'll talk about here in a minute. So this is something that is very short-lived. So it'll grasp our attention, it'll orient us to it, and then we kind of go back to voluntary control. And that's an important evolutionary adaptation, right? We don't want these reflexive things capturing our attention for a really long time, because then it's way too distracting and we never get anything done. So researchers, this is just something that I'm putting up here to kind of explain these next couple of tasks that we'll talk about. They use reflexive cueing. So it's something that's a distractor, something that captures your attention that's not part of your goals. And that's going to be important for the couple of experiments that we'll look at here in a minute. I may skip a slide or two. I want to show you guys neglect. So inhibition and return, I'm going to skip through this really quick because I'll talk about it again on this next slide. It's just this idea that if you guys have ever done a Where's Waldo, and you're searching this whole page, right? And let's say Waldo's up in the top left, and you look in the top left first, and you're looking for Waldo, and you don't find him, you leave the top left. you don't return to that space because your brain is like, I already searched that space, it's done, it's over, and you move on to search all the other stuff. And then you get pissed at yourself and you come back at the very end and it's been there the whole time. So our brain has this mechanism where if we've already attended to something and decided that that area of space is not important, our brain won't return to it. It's this inhibition of return. And so I'm going to skip over this. This was just a study that kind of showed that if we presented something before 300 milliseconds, then they looked at it. If we presented it after 300 milliseconds, then they weren't attending to it anymore. And so it's important in visual search. That's what I wanted to kind of get to. So you guys have been at airports before. You have the baggage coming off the carousel, and like you were dumb enough to get a black suitcase. It's medium size. It looks like everybody else's. If you were smart enough, you got like a bright pink one that like sticks out, right? That's kind of this idea that's going on over here. So this was what I was just talking about with inhibition of return. So our gaze is biased towards new locations. So if we've already looked for something in a certain location, we're less likely to look again in that area. And getting back to kind of the idea of the baggage. So items are really easy to identify if they have salient features. If you have a bright pink bag, it sticks out, right? So this red circle amidst these green X's and O's is really, really easy to see. And it doesn't matter how many more green X's and O's I add to this, it's going to take just as long to find that thing. It's called pop-out search. This has really salient features that are able to direct our gaze really, really easily. Now, if that bag is black, like everybody else is, it's medium size, there's no distinguishing features on it, then we're in this field right here. It's a conjunction search. And it's this idea that if we add more distractors, so if there's a ton of bags on the carousel, then our time to find it is going to increase linearly. So the more distractors we add, the harder it's going to be to find that thing. And they think it's because we have these attention mechanisms that have to actually search and check every individual thing. And so if I tell you to find the red circle in this one on the right, it's a lot harder to do when there's other things that share features with it. You have to look at the spatial features that are associated with it. It doesn't just pop out at you. It's right there. So reaction time is increasing. The more stuff you add, the more distractors you add. So this is just demonstrating that this also happens for features. It's not just for simple spatial type stuff, because everything we've looked at so far has been talking about regions of space. This next one is actually a better example of this, so we're running out of time. This one was just saying that, like, if I tell you to look here, this field of space is also going to be boosted because it's part of the same object. And so these higher visual areas notice that even though I'm supposed to look right here, this thing is a coherent whole thing. And so I need to boost signals across the entire thing. I've recognized that that's an entire object. That's kind of what this is getting at down here. Even though it's not in the space that I'm attending to, it's part of the same thing. And so I need to attend to that entire chunk of space instead of just that little spot. And this was the experiment that really worked a lot of that out. So this was Nancy Canwisher. She was really big in a lot of the face processing stuff that we talked about with object recognition. And so they scanned these people while they were looking at these pictures that had a face and a house kind of overlapped with one another. And they directed their attention either to the face or the house. And we know from what we talked about with object recognition, fusiform face area is what is tracking face perception. And the parahippocampal place area is what's tracking scenes, is what's tracking the house. And so what they saw was that if the attention was directed to the face, there was a boosted signal in the fusiform face area. And if the attention was directed at the house, there was a boosted signal in the parahippocampal place area. And so this is just kind of beating a dead horse at this point of just kind of getting the point across that we're having these signals are being boosted for things that we're interested in. And they even told them to pay attention to movement, so not to pay attention to a face or a house, and then they moved the face or the house. And even though they weren't specifically told to pay attention to a house or a face, because they were attending to a face that was moving, that's boosted the face area as well. And if they were paying attention to movement and it was a house that was moving, then that's boosted the signal in the place area. Cool. So this kind of ties a lot of that together. So this is kind of the last thing that we'll talk about as far as anatomy goes before I introduce neglect. And this is what really ties into what we talked about with the networks with ventral stream and dorsal stream in object recognition. So you have this dorsal stream that is really important for voluntary control. So this is the one that's kind of going over the top. It includes these frontal lobe regions that have been implicated a lot in these attentional control mechanisms. And when we get into memory, you'll see why, because the frontal lobe is really, really involved in working memory, in short-term memory. And so the frontal lobe is keeping online all of these things that it's interested in, all of these goals and intentions, and then it's using that spatial and working memory to then send signals back down, like this is what we need to attend to next, this is what we need to attend to next. And what they're seeing, this is kind of what I was hinting at with Lewis, is they've done these studies that show that these regions in the frontal lobe will actually sync up their firing with these regions, these early processing regions, and they'll talk to them. And because there's firing coming from here to here, that's what's actually boosting the signal. And they can see this on EEG. They'll see that this area and this area are firing together. They see that on like resting state type stuff. So what I want you to take from this is that the dorsal network is the one that's involved in the voluntary control, and the ventral network is the one that's more important for reflexive type stuff, orienting to salient distractors. So it uses a lot of the information from that other network, but then it shuts down those voluntary controls, and it automatically orients things. So that's really what I want you to take from these two, is that dorsal network is the one that's doing a lot of the top-down processing, and ventral network is the one that is shutting down that top-down stuff in order to orient to things that are important. Evolved from necessary survival type behaviors. So we'll look now at failures in attention. This picture really kind of strikes a chord with me. My wife at 21 weeks pregnant was in this situation, and her car was flipped by someone running a red light. Like, I gotta tell you, like, my son has had the most epic pregnancy in the world. Yeah, I'm never buying anything but Toyota again, because like the car was just like a heap of metal, and every seat inside is like perfectly intact. So please don't look at your phone, because from what we learned from this lecture is that we can only attend to one thing at a time. Even if you think that you can drive while texting on your phone, it's not true, because you're not looking up, and you're not attending. First thing we'll talk about is, this is just kind of briefly, is attention deficit hyperactivity disorder. So this is one that has popped up a lot in the current decade. Chronic condition, it's marked by inattention. That's why I'm bringing it up. They have a really hard time paying attention to things. So it's like this filtering process isn't working, and there's just a ton of different distractions. So it may be that the reflexive attention network's working really, really well, but the voluntary one is not doing so hot. And there's some early work that's suggesting that there's actually decreases in white matter in these voluntary attention networks. So this is very early work. I haven't seen studies that have enough subjects to really make a positive distinction either way, but this is an interesting idea, that there's less wiring in people that have this condition. that prevents the voluntary control from having the type of control it has. Yeah. So neglect, this is that picture I was telling you about. Crazy. These people are not aware that they have this disorder. And so it's this complete lack of attention to an entire hemisphere. So this is usually due to some kind of stroke that has caused specific lesions in the attention network in one hemisphere. And like I hinted at, they have no awareness that they're doing this. They've just completely ignored the fact that this side of their face is uncounted. So they'll look in the mirror and they'll only, their eye gaze and everything will only pay attention to this side. And there's some tests I'll show you in a minute to kind of get it at this. So that's what I really want you to take from this, is that neglect is kind of this ignoring one side of our visual field and one side of our body. This was really, really interesting. So this was a man that was an artist that had a stroke, and it affected half of his attention network, and he started to do self-portraits. And so this was a portrait that he drew of himself right after the stroke. And you'll notice that there is just absolutely no left side of the body that he drew. It's just like as though it didn't even exist. And as he worked through this, and we've seen this with some subjects or with some patients, that they can kind of retrain their brain to regain some of the function. And so this was over time after the stroke. And so you see that here it's starting to get better, but it is still very, very concentrated on that right side. Here you're starting to get some left features included. And then I can't remember what the date was on the first one and how long this had been, but by this point he was finally starting to include some of that neglected hemisphere. And we'll see on the slide at the end that they're not blind to these areas, they're just not paying attention to them. And so that's something that could be contributing to this fact that he's aware that he has a disorder and he's trying to attend to things that his brain is trying to tell him not to. So this one was a study that looked at eye movement. So they were having them search for a specific target. This is a control subject, and so the control subject mainly kind of focused on this middle region, but searched the entire visual field. Whereas this person with neglect, their eyes only searched the right half of the visual field. Like, this just kind of blows my mind. Like, really, really interesting disorder. And these tests were developed, and a lot of the times, if you end up with brain damage, or if you end up in a car accident or something, you'll have psychologists that will come and give you these tests to see if your attention networks have been affected. So this first one is a line bisection test. So they give you all of these lines on paper and they tell you to draw a line through all of the ones that you see. And this one, you'll notice even the ones on the left, the lines that bisect them tend to be kind of further to the right. And then you have these ones that are completely missed. This one, though, is one that's really, really common that you'll see a lot. My wife's cousin actually got in a bad car accident 2 or 3 months ago. And right after the car accident, I saw a clock like this. And my wife's mom was like, "Why are they having her draw a clock? People this day don't know how to read an analog clock." I don't think they're asking her to tell time. Yeah. So at first it did look like this. And so they're actually, they know that all this stuff should be included, but when they draw this clock, they're not including any of the stuff on this side. And they think that it's done. When you ask them if they're done drawing the clock, they're like, yeah, it's all there. It looked like this. Her cousins looked like this at first, and now it's a lot better. So these things can be overcome to a certain extent. This one was really cool. So this guy in Milan, in Italy, collected a bunch of neglect patients. So these patients had all been to these areas that we're talking about, and he told them to just imagine that they were looking at the Milan Cathedral and describe what they saw. And so this was just in their imagination. They were just pretending like they were looking at it. They described only the things on the right. And then they said, okay, now turn around in your imagination and look at the Duomo and describe everything you see. And same thing, they only described the things on the right. The crazy thing was that even though they were describing things on the right, the things on the right here were the things that they left out when they were looking this way. So really interesting that this is something that's not just like visual input. This is also happening in things that we're trying to conjure up in our imagination. Really, really interesting. So they failed to describe what was in those missing fields, even though that stuff was included when they're turned around the other way. And last slide, extinction. So this is just to get the point across that these patients are not blind. So if you show them the comb and you show them the spoon, they can see both of those. The problem happens when there's competition. So if they're holding two things, then it biases them towards one or the other. And it biases them towards the side that they're usually paying attention to. So this guy, if I concentrate, I will see it. But if there's any distraction, I won't. And I think that's kind of what gets at that person being able to draw the entire self-portrait, is that he was able to kind of limitate the distractions and focus and concentrate. So that's it for today.